Yes, thank you very much for the kind introduction, and thanks to MS Vision for having me here today. And yes, so in the next 50 minutes I'd like to show you how I made use of native MS to study subunit exchange and protein complexes, and how we use modeling to even dig deeper into this kind of data. So when we think about a protein complex doing structural analysis, one thinks that this protein complex is rather stable, since for most methods like cryo-EM it has to be stable. If it's not stable, you will not see it in most cases. But in action, we have to think about a protein complex as a rather dynamic assembly of different proteins. So there is protein subunit exchange happen. There are possible roles for this subunit exchange, like a dual localization, which is known from the nuclear pore complex, or it can have regulatory control. This is known for tight junctions, and a repair mechanism like in ribosomes, where one subunit that's broken will be exchanged by a new one which is intact. Furthermore, it can be a functional adaptation as known from the spliceosome. The subunit exchange is known that it happens on a time scale from seconds to even over years, so this is quite different. And in my project, we are asking if this protein subunit exchange also plays a role in the peroxisomal protein import. A few words about the peroxisomal protein import. I had a talk yesterday in the structural biology symposium about the cycling receptor model of the peroxisomal protein import, and rather quick, it's worth to mention. Okay, so that we have a docking event here, where the cargo protein is then handed over to import pore, where the cargo proteins, which are folded or even oligomeric, are translocated into the peroxisomal matrix. In my study, we combined methods from native MS, cross-link MS, and cryo-EM to come up with a following model shown here on the right side, where we have a heterotetramer of PEX14 and 17 sitting in the membrane, and the pore is mainly formed by the receptor protein PEX5. This model is nice to see if we compare the known endogenous and artificial cargoes that are shown to be imported into peroxisomes. So if we have a look on the enzyme MDH3, we have a size of about 5 to 9 nanometers, and then there's a study from 2018 from Young et al. Which describes peroxisome even as omnivirus, since this cargo was even larger, so it's a factor of about 2, and everything was imported into peroxisomes. And from this we can deduce that this import pore and also the docking complex on the left side has to be rather dynamic, since to the different sizes of these. Okay, what is this? Can I just delete it? Okay. All right. The docking complex has to be dynamic, since we have these different sizes of the cargo proteins. Yeah, and by native MS that we are using, it's a water Synapse G1 customized by MS Vision, and we have several modifications on this instrument, and the first one is an open and nano-ESI source, which helped us with a thermal control on the nano-ESI source. Furthermore, we have a sleeve on the traveling wave ion guide in the source region, which helps us to have a more uniform pressure gradient. And we have an advanced gas handling for the trap, IMS, and transfer cells. So we can flush all the 3 cells with collision gas and adjust the pressure for each cell. And in sum, all these modifications improve the transition of labile non-covalent complexes. So to come again to the subunit exchange, I will show you now the general workflow that I'm using, and it starts with a sample preparation where we need 2 mass different versions of the same protein, so a heavy and a light variant. These 2 versions will be mixed and then directly transferred to the electrospray ionization in the source region. And after you acquired your mass spectrum, you then have to perform a chromatogram parsing. For this I'm using UNIDEC. And after the chromatogram parsing, you can do a data extraction and the extracted data can then be analyzed using kinetic modeling, where you then monitor the relative abundance of homo-oligomers and formed hetero-oligomers over time. And by this, you get an idea about the dynamic of a complex in its, regarding assembly and disassembly processes. So and now in the next slides, I will just guide you through this workflow. And regarding the sample preparation, there are different strategies in the field how to generate these mass different species. And one of the first papers that came out in 2002 by Sauer and al., they used orthologs from different species. And the advantage of this is that you have fast sample preparation, and a disadvantage can be that you have a potential high bias due to high variant in sequence, or there is even no ortholog for the protein of interest. Besides this, of course, we can do isotopic labeling, and the advantage of this is that you have identical physical properties, and the disadvantage is that it can be quite costly and that sample preparation is more sophisticated. The strategy that I'm using is a cleavable affinity tag, so the protein that I'm investigating are recombinantly expressed and purified, and I'm using a TEF cleavage site just to cleave off the tag, and by this I have a portion with tag and the other without, and then I have two mass different species. Regarding the actual time-resolved native MS, we have to distinguish two different strategies. The one is a discontinuous sampling, which is mostly done by this Advian Traversal Nanomate robot, and the continuous sampling that I'm using. What is this robot actually doing? So you have a 96 well plate, and in this you start the reaction of mixing, and then the robot picks the sample and transfers it directly to the EASY. By this, the robot has a delay time of 32 seconds, and for each data point, he starts this again and again, the process. So the reaction takes place in the plate where you have a good thermal control, and then he needs always 32 seconds to use a new needle and start an acquisition. In the continuous sampling, this is different. When I mix my sample by pipetting, I need 55 seconds roughly to fill my needle and start the acquisition. And after this, I just run the measurement for about 20 minutes or even longer, depending on your reaction. And then I pass my chromatograms for each data point. So to sum this up, we have for the different strategies, different advantages. The robot has a higher reproducibility, it's automated, and the temperature control is even more accurate. The continuous sampling, in this way I'm doing it, it's inexpensive, you need a short training period, and you have a higher flexibility of the experimental design. The disadvantage is the robot is costly and requires longer training period, and the continuous sampling, it's less reproducible, and you may have potential pH effects, since one needle is for about 20 minutes or 30 minutes in front of the source and under a high potential, right? If you then acquired your mass spectrum, your measurement, you first have to start with the conversion of the raw file in the Waters format, in my case, to MZML, to then go to UNIDEC for the chromatogram parsing. Within UNIDEC, I'm using the latest version, 5.01. You can do the chromatogram parsing by the HDF5 import wizard, shown here on the right. And within the import wizard, it's probably a bit small. You then load your raw file, and I'm using the scans option, where you then slice your raw file. In this case, every 5 scans become one single mass spectrum. Then you hit the load all to HDF file button, and this HDF5 file can then be loaded in MetaUnidec, shown here at the top. Within MetaUnidec, you then have, as I said, for each time point, here, so starting at 0 to 50 time points, a single mass spectrum. On the right side, you then select your processing parameters and the deconvolution parameters, as well as the picking for the different species. After you select your needed parameters, you then hit the Run All button here at the top, and you will receive the deconvoluted spectra over time and the identified species with the intensity. The peak integration will then take place at the data export in a CSV file, and the CSV file can then be handed over to the kinetic modeling. In my experiment, I analyzed the PEX14 trimeric version. Since we have a light and a heavy copy, we have 6 different species. After mixing, we have in total 9 observable species of the reaction. And we can have a hypothesis about this reaction. So in the first case, it could be a decomposition of trimers into dimers and monomers, followed by reassociation into trimers, or it could be a collision encounter process. And to answer this question, which of the hypotheses is more true, we teamed up with Hossein Yamanadi from the Timmers group at the University of Freiburg. And they established a protocol or program called Data2Dynamics, which runs in MATLAB. And he formulated for this reaction the different reaction shown on the left side. And the first set of reactions account for the buildup and decomposition of homo and heterotetrameric dimers. The next 4 reactions stand for the buildup and decomposition of homomeric trimers, and the last 4 are for the heteromeric trimers. So we have in total 16 possible reactions, which are considered in the model. The results look like shown here. So for each species, we then have the experimental data denoted as stars, the fit as the black line, and the confidence interval for the fit in gray. We have a full set of species that have a low intensity in the mass spectrum and therefore become a larger confidence interval by the fit. But we also have, especially for the trimers, a quite good model about the abundance over time. I now like to focus on the behavior of the light homo trimer, which incorporated one heavy variant, where we have in the native MS data, we can only deduce the relative abundance of this species over time. But if we now use the model, we can generate so-called flux plots. In this flux plot, we have the contribution to the ABB flux of each reaction here on the y-axis, and the net flux here as a dotted line. So when we look here, the relative abundance, which changed at the very beginning quite fast, we do not know which reaction is responsible for this behavior here. But in the flux plot, we can see that the association of a monomeric A with a dimeric BB mainly contributes to the flux of the species at the beginning, which becomes less dominant after about 500 seconds, where then this reaction of the mixed dimer associated with one monomeric B becomes more dominant. And the decomposition of this trimer into the dimeric BB with 1A will also increase over time, and then after about 1000 seconds we have a net flux of 0, so the reaction is in some kind of equilibrium. So the sequential reactions can be visualized nicely by these flux plots, and this would be not possible without the modeling. Furthermore, this Data2Dynamics software is able to draw this kind of interaction schemes, where then half-lives for each of these arrows can be calculated. Since this project is preliminary data, I'm not able to show you numbers for this today. But how can we translate this information into a biological question now? So I started with a current model of the import complex here on the left side, and by the use of the monitoring of the subunit exchange, I now draw the hypothesis that depending on the size of the cargo, there is probably an assembly of a second copy of the heterotetramer, which is then some kind of a functional adaptation, or if one of these copies of the PEX14 is broken or is too old or whatever, it can be also displaced, and then a new copy can be assembled inside. Yeah, and if you assume that this heterotetramer is intact, there's additional data about the Daniel 2, which I did not include in the talk here today, but yeah, this can be bridged, and depending on the size, then a large cargo together with the receptor PEX5 is then guided to the membrane, and after the receptor build up the pore, the cargo is then translocated into the peroxisome. Yeah, and with this, I like to come to my acknowledgements. So the whole Warscheid group, especially Bettina Warscheid and Friedel Drepper, who was involved in the modeling together in the collaboration with the Timmer group from the Physics Institute at the University of Freiburg, And I'd like to thank my graduate school, RTG, and especially MS Vision for the modifications on our instrument, and you for your attention, and I'm happy to take your questions.